Okay, so now we are back with translation and you got some of the behind the scenes. So now we're actually going to talk about how does this occur. Just like in transcription, there's 3 stages of translation and they actually have the same name, so it'll be easy for you to remember. Those stages are initiation, elongation, and termination. All 3 of these stages require protein factors, which are essentially different enzymes that help catalyze the translation process and put everything together. And the energy for this process is provided primarily by the hydrolysis of a molecule known as GTP, which stands for guanosine triphosphate. It's very similar to ATP, except for instead of adenine in there, it's guanine. So if you see GTP, I know we've briefly mentioned it before, that's what is being used to catalyze and give the energy for this process. So the initiation stage is the first stage of translation, and this is when the small ribosomal subunit binds with the mRNA and the special initiator tRNA. And the small subunit will move along the mRNA until it reaches what's known as the start codon, and this is AUG. And if you remember from the transcription section, AUG actually is the code for the methionine amino acid, but it's also the start codon. And this signals the start of translation. Once this has occurred, the small ribosomal subunit binds to the mRNA, and once it finds the start codon, the large ribosomal subunit, the other piece, comes in and binds and attaches to the small subunit as well as the tRNA and the mRNA, and this forms what's known as the transcription initiation complex. And once again, in order for these two to come together, we require the help of some protein factors, for help with translation, and also the hydrolysis of GTP helps get this transcription initiation process made. So at the end of initiation, the first tRNA can be found at the P site, and the A site is available and waiting for the next tRNA to come. So you can see that this tRNA, once again, has that code for the amino acid methionine. So that's why that's the one that's up here. And now the A site is waiting for the next amino acid to come. And then here's our mRNA with this set of 3 being a codon that's complementary to this set of 3, which you can see as an example right here. AUG is methionine, and then the anticodon is the corresponding complementary base pairs. The start codon is very important because it establishes the reading frame for the mRNA, and then once that large ribosomal subunit is added, that's when the translation initiation complex is complete. So the next step of translation is known as elongation, and during this stage, like you can imagine, the polypeptide chain is getting longer. So amino acids get added one by one to the previous amino acid at the C terminus of the growing chain. Now, this requires you to go back and think about Unit 1, where we learned about the structure of the amino acid. Remember, they're composed of both an amino group and a carboxyl group, and so the C terminus is the side with the carboxyl group. In these pictures, you're not really going to see that, but it is important information. So how does this exactly work? We have kind of 3 steps within the elongation stage, and once again, these steps are catalyzed and helped along by some protein factors known as elongation factors. And this proceeds along the mRNA in the 5' to 3' direction. So the first step is known as codon recognition, and this is when the tRNA comes in to the A site that has the complementary anticodon to the codon of mRNA, so that this tRNA arrives and is available to take the place in the open A site. The next step is known as peptide bond formation, and in this step, what happens is like the arrival of the tRNA in step 1 catalyzes several reactions. So one is it breaks the bond between methionine and the RNA in the P site. And it also allows a peptide bond to form between the methionine attached here and to the amino acid attached to the tRNA in the A site. And then finally, we have what's known as translocation. So you can see here the peptide bond is forming between these two, and so the amino acid chain gets added to this tRNA, and now this tRNA doesn't have any amino acids attached to it. So in the next step of elongation, this tRNA gets moved as the ribosome moves. This tRNA moves to the E site, and so finally the empty tRNA is in the E site, which if you remember is the exit site, and so he leaves and goes back into the cytoplasm to pick up hopefully another amino acid. And so just once again, I'll review this one because this is probably the most confusing step. In the first part, a complementary anticodon tRNA arrives at the A site, and these bases pair up with the codon of the mRNA. This causes two things to happen. So one is the bond between methionine and the tRNA is broken, and that's the one in the P site. And then a peptide bond is formed between the methionine and the attached amino acid in the A site. And then that ends up adding the attached amino acid to the polypeptide chain. Now the tRNA in the P site is empty, so the ribosome moves over, and that empty tRNA moves to the exit site where it is released back. So elongation will continue to occur, and we keep getting a longer polypeptide chain until termination. Termination is signaled when the mRNA reaches the stop codon at the A site of the ribosome. So at that point, the stop codon is either UAG, UAA, or UGA, and that signals the arrival of a protein known as a release factor. So in this case, once the release factor is bound to the mRNA, hydrolysis occurs. And if you remember, hydrolysis is the addition of a water molecule that can break a bond. And so instead of adding an amino acid, this adds water, and the adding of water breaks the bond between the polypeptide and the tRNA, allowing it to be released, and then the rest of the translation assembly can come apart. So here would be in the termination step of translation, we now have the stop codon is coded for, so either UAG, UAA, or UGA, and then the release factor binds instead of another tRNA. That release factor promotes hydrolysis, so it cuts the polypeptide free from the tRNA, and then everything else is signaled to come apart after that point. So one important thing to note is that once translation is finished, that sums up translation, what we just finished. So initiation, elongation, and termination. But once that's done, typically the proteins are still not ready to be functional, and they get modified additionally after they have been made via translation. So one thing that has to happen is the polypeptide chain has to coil up into its 3D shape. So remember, it goes through several different types of modification to get to its 3D structure. And then additionally, in order to do their jobs, most proteins have to require some modification. And this is because when the proteins are made, almost all of them are made by free ribosomes in the cytoplasm, and they are the ribosomes in the cytoplasm is kind of like the default location to make proteins. And it can fold into a 3D shape and remain in the cytoplasm, but a lot of the time we need proteins in other areas of the cell, and so they can be moved to another location when they are marked by something known as a signal sequence or signal peptide. And so in this case, the signal sequence binds to the signal peptide, and that kind of tells, it's like a short stretch of amino acids that tells where a polypeptide or protein belongs and tells it where to go. So if there is no signal sequence, then the protein will stay in the cytosol where it was formed. But if there is a signal sequence, that will tell it to go somewhere else, for example, the endoplasmic reticulum. And then there are also a lot of other modifications that can happen to proteins once they've been released from the cell, and these include the addition of lipids, sugars, phosphate groups, etc. So that's a little bit more about what happens to the protein after it's been translated. So we'll stop this video here, and then the last section we have to talk about is mutations.